Hello, this is your kind of video for the fifth lab number 5, photosynthesis. And if you start by reading the background, you need to understand this major equation for the lab because it explains why the leaf discs that we are going to use will float. One of your substrates, or the main substrate here, that we're going to add to this experiment is sodium bicarbonate. Sodium bicarbonate is responsible for providing the solution with free carbon dioxide that the leaf discs will then use as they do photosynthesis. All right, and what I'm going to go ahead and show you here is just the main setup that you will then modify depending on which independent variable you get assigned. So the basic setup is that you will have these containers of bicarbonate. We are going to use a 0.2% sodium bicarbonate solution, and those again are already labeled for you at your lab benches. And the first thing you are going to do is you are going to pour the sodium bicarbonate into your cups. You've got 3 plastic cups. You might not be pouring this into every cup because if you are, for instance, this group, and you are manipulating bicarbonate concentration, you can see that these guys will end up having a 0.05, and then we'll also have a 0% sodium bicarbonate. So you are going to have to kind of pay attention to what your individual experimental conditions are. All right. But assuming you are going to use all 0.2% bicarbonate, you can go ahead and add them to all your cups. And we are not gonna measure this necessarily. Just go ahead and eyeball it. You want about 200 milliliters. It gets you about 3 centimeters worth. I'd say no more than that. What I have in the cup is plenty. And that's step 1 in your procedure. The second step in your procedure is an important one, but it is easy to skip. Do not forget to add these 5 drops of soap. So you will also see soap in your green setup trays. You're going to go ahead and add 5 drops of that. Again, to each one of these 3 plastic cups. I'm just going to go ahead and do it to this one, and I'm only going to use this one in the video just to make it a little bit simpler. This is a surfactant. It will help the discs behave the way that they're supposed to in the lab. Sinking them. Yes, sinking them. The soap wets the hydrophobic surface of the leaf, allowing the solution to penetrate and sink the leaf. Sorry, I forgot for a second. Okay. The third thing you're going to do, this is a paper leaf, but you are going to have actual ivy or some other kind of leaf, depending on whatever year you're watching this video in. But imagine that this is an actual leaf. You're going to hole punch, using your hole punchers, 10 discs from these leaves onto a sheet of paper. So imagine this is a white sheet of paper on your lab bench. Hole punch directly onto there so that you can find them and you're not trying to, dig around for them if they're contrasting on the back of the black lab bench. But let me pick you up. You can see that you will have, you'll have 10 of these little discs. I only have 1 little disc right here. But try to avoid hole punching the veins. So these are the veins on the leaf. You don't want to get that. That's xylem and phloem tissue. You kind of want to hole punch off to the side. So you would hole punch like all around in here. Try to get as many holes per leaf as you can. And again, you want 10 per cup, which means you're gonna want 30 of these total. All right, so after you do that, step 4 is to take one of your syringes and you are going to, I would recommend, kind of hot dog folding the piece of paper that has the hole punches on them and empty your little discs into the container. So you can kind of see my 1 little disc that I've done. You're gonna have 10 in here. But then you're gonna take your stopper for the syringe and you are going to push come on. You're gonna push the syringe all the way up until about here. You're gonna leave a little gap here because you don't wanna crush the leaves. So you're gonna have a little air pocket. And then you're gonna take your sodium bicarbonate solution, and I would recommend resting your syringes adjacent or next to the cup that they are gonna be used for, because not all 3 cups will be the same. So that way you're not mixing and matching which cup you're using with each syringe. And you are going to suck up about 4 milliliters of solution, bicarbonate solution. And here's the tricky part. At this point, you need to get all of the air out of the spongy mesophyll in the leaf discs. And you're gonna do that by creating a vacuum. And it's described in your setup, but basically, you're gonna push all of the air out to the best of your ability. Put your finger or your thumb on the top. I'll put my index finger on the top. And then pull back on the plunger, keeping your finger on the tip, making a vacuum, and it'll resist. It's not gonna wanna actually pull back nice and easy. So you've kind of gotta put some effort into it, shake it a little bit, maybe jostle it around, and that will kind of suck the air out of the spongy mesophyll, and it will replace it with the bicarbonate solution. So you might have to do that a couple times. You can let go of the syringe every now and then to kind of reset and kind of start again if some discs are not syncing. But the basic idea is that you've got to get the discs to sync, and you can see it's sunk to the bottom of my syringe. You're gonna want all 10 discs. And I can help with this, and if other people are good at it and you're struggling with it, then you can always ask for assistance. All right. At this point, when all 10 discs have sunk, you can just take the matching container and just dump them right out into the cup. And what's gonna happen is all 10 discs will sync just like this. And then if you follow me, you will place them. I'll have these trays set up around the room on these little plant growing stands. And you are going to count the number of floating discs that rise to the surface every couple of minutes. And you've got space in your lab to record that data. And what you're gonna see now, if you go to the data collection page, the data table page, is that there are 4 different groups. So if you did the group that I just did, and you used my 3 cups, you're gonna manipulate the concentration of bicarbonate. So that's why this group has a 0% bicarbonate, or a 0.05%, and a pure water, a 0%. All right. If you're the group that is doing pH, then you are going to use these sodium bicarbonate pH buffer solutions. So you'll use pH 3 and pH 11. If you're one of the groups doing temperature, then you will be placing your cups either on ice for cold, you're gonna leave them out for room temperature, or I'll give you boiled sodium bicarbonate solution for hot. All right. Finally, the last groups are the groups that are gonna do distance, so light intensity. If you're a light intensity group, you're not gonna put your cups over in the trays at all. Your station is gonna have lamps over here that you'll go ahead and turn on, and you'll put your 3 cups at different distances to manipulate light intensity. So I hope this helped. That's the basic setup, and I'll see you in lab.